So I will, thank you again. My name's Olive. I'm from Stony Brook University. I'm a graduate student. And I'm going to be giving you guys a little bit of a break from lunar research. And I'm going to be showing you some preliminary results that I've collected from some nano-IR analysis using of Ryugu samples, nano-IR analysis of Ryugu samples. So, oops. All right. So quick background on Ryugu and why it's interesting. So Ryugu is a near Earth asteroid that has a lot of primitive materials that studying it can tell us about the early solar system. We believe that Nano-IR instrument is particularly useful for Ryugu analysis because its ability to characterize hydration states. And because it's known for being non-invasive and provides high spatial resolution of these extremely small particles. So basically, just what I'm going to be showing you is the nano-IR analysis we've done so far, and kind of how we've done that is we've taken maps of regions of interest and then select points on those maps to collect point spectra. And our goal was to target large phosphate grains, the phyllosilicate matrix material, and then other notable grains like carbonates and phosphides. So to the left here is an image of what we kind of see when we're using the nano-IR instrument. So out of focus, we kind of have this arrow that on that arrow is this AFM tip that comes within a wavelength of light to the sample and allows us to collect our spectrum. And so here we can see this is one of our Ryugu samples. And so I'd like to just point out this grain that the arrow is kind of pointing to. So I'm going to show you our first map that we've taken, and you can kind of align that with the white grain that you kind of see at the bottom of that map. So we're looking in that region there. And on that last image you saw, it was pretty much this kind of dark gray background with a couple notable like white grains on there. But when we're looking at these maps in the Nano-IR, we can see there's actually a lot of variety of things in there. And these maps are 40 by 40 microns in size. So there are these very small regions. And as you can see, there's a lot of notable things within that. So we have these point spectra that we've selected different regions on this map. And so you can see each of the spectrum on this figure to the right that are color-coded with the points on the map to the left. So I'm just going to kind of highlight what we're seeing in some of these maps, and I'll go through a couple different samples that we've looked at. So as you can see, the blue and the yellow spectrum we see here don't really show anything notable. So some further analysis, either in different wavelength ranges or with different instruments, will have to be done in those regions to identify what we're looking at. But we do notice the red and purple, so 2 and 4 on this map here, have a very noticeable feature around 1100 wave number there. Which that range tells us that it could possibly be phosphate or sulfate. This kind of doublet feature we're seeing there is pretty interesting. So to help us identify that further, we're going to be doing some high-res mapping, SEM and EDS mapping of those regions to kind of identify what we're looking at there. And then the green, there is a kind of notable feature between that 10 and 1100 wave number there, which could be phyllosilicate matrix, kind of that silicate peak there. So here's another map that we've taken in that same sample, but a different region on that sample. And I wanted to highlight this map because we're kind of seeing some different things spectrally here. So the red, the yellow, and the purple, so 2, 4, and 3, those sites, we're not really seeing anything noticeable spectrally. But 5 and 1, the green and blue spectrums we're seeing here have very noticeable peaks between 14 and 1500. that lead us to think those are likely carbonates. So within the same sample, we're seeing a few different things. And this is a different sample here. This is a microtome slice here, and on the left we have an SEM map that we're looking at. And I have this box kind of highlighting the region that we're looking at, the Nano-IR, with some triangles that are color-coded with each of the spectrum shown here. And first, we have the blue on the bottom here that again has that feature between 10 and 1100. That's likely silicate. So we think that would be phyllosilicate matrix there. The green and purple, the two spectra at the top here, don't really show us anything notable. So again, we'll probably have to do some further analysis on those regions. But what's particularly interesting and why I wanted to show this samples analysis is the red and yellow spectra we see here that are both on the same notable grain here. The yellow spectra looks a lot like what we would expect from an apatite. There's this notable kind of triplet feature that we're kind of seeing to the left of the peak at 1100 wave numbers there. But the red, however, looks very different spectrally. And so even though those were both taken on that same grain, we can kind of conclude that Ryugu is very heterogeneous. So you will have to really do some analysis to see really how heterogeneous that is.
3 minutes.
So I'm just going to kind of wrap up and then talk a little bit about some of our future work we plan to do with this. But so basically, as I said before, Ryugu is extremely heterogeneous in nature. And we've seen some evidence that there's phyllosilicate matrix, of course, as well as phosphate grains, carbonates, and evidence for phosphides, sulfates, sulfides, and magnetite. And then some future work that we hope to do is to use Nano-IR to look in near IR wavelengths to help further identify some of these materials, as well as, I mentioned, doing high-resolution SEM and EDS mapping to help identify some of these features that we couldn't quite identify with the Nano-IR. We also have an algorithm that we are developing to model far field spectra using near field data that we can compare with remote sensing data. And so that's the really exciting part of this work. So a lot of this is preliminary work for that comparison. And then as well, one of the big ticket items people have been talking about nowadays is the Bennu samples. So being able to compare this Ryugu analysis to Bennu samples would be very exciting in the future. So thank you so much for having me. And I'm happy to answer any questions.
All right, we have about a minute and a half. We could probably take a question or two. So anyone?
I'll ask one. So for your future work, do you need to expand on your samples that you already have? And I understand that you want to do more work on the samples that you're already having, but do you need to branch out into all kinds of different samples you can get your hands on from Ryugu, or is what you have enough?
Well, so right now we're still in the process of collecting this data. Some of these data that I showed today are actually ones that we just collected within the last couple of weeks. So we have quite a few samples that we're still going through and analyzing. And even within these samples, there are lots of regions within these samples that I still have not really gotten a chance to look at. Obviously, the more data, the better. So I'm going to be trying to do as much analysis as I can on these, as well as trying to look in as many wavelengths as possible to kind of give a broad analysis on these samples.